Okay, second to last video of the unit. There have been a lot this unit. It's a very lecture-heavy unit. I apologize, but also not really, because I like talking about it. This one's on mutations. Kind of been ignoring them and avoiding them this whole year. We will finally talk about them and their role, their purpose, a little bit. So, what are mutations? They are just changes in the genetic material of a cell or a virus. It could be harmful, beneficial, or it might not do anything at all. We can split them kind of into two types. Somatic mutations are in our body cells, so everything but the gamete-producing cells, the ovaries and the testes. They are not passed down because of that. They're just in our body cells. They're in our skin cells, for example. That doesn't get passed down. But germline mutations occur in the cells that give rise to sperm and egg and are passed down. So mutations that happen in the germline are considered more serious because they become essentially permanent and get passed down and enter the entire human gene pool. So let's talk about some specifics. There's a few major classifications of mutations where we'll talk about the most basic first, and that is point mutations, which are just mutations that happen at a single point, single base pair. And there are two main types, being either substitution or an insertion/deletion. A big example of a point mutation is sickle cell anemia. That is a single base pair change from a T to an A, which leads to a different amino acid being put in, which causes the whole thing to fold differently and causes all the issues that sickle cell anemia has. So, substitutions. This is what sickle cell anemia was, or is. This is generally the type that is most likely to have no effect. You're just replacing one nucleotide and its complementary pair with a different pair. So they're still correctly base pairing, it's just that you've changed the actual sequence. You've changed from a C to a U, as shown in this example. And this can have the kind of three different effects depending on how bad it is. So the first are synonymous or silent mutations. Basically, the mutation changes and it still codes for the same amino acid. So no harm, no foul. That's seen in the upper one here, where it doesn't matter if it's a U or a C at the end, it's still coding for glycine. This is a feature of the redundancy of the genetic code. A benefit of it is that it kind of does give us this resistance to mutations, because if I make a change, especially to the third letter in the sequence, it's almost very few times it'll have a serious problem. Missense mutations are ones that do change what amino acid it codes for. So often these will be in the first DNA or RNA nucleotide in the codon. If it changes the first letter in a codon, it's almost certainly going to change the amino acid. Generally, changing the first and second will change the amino acid. Changing the third, you only have like a 40 to 50% chance of actually changing it. So missense can be a problem, changes a single amino acid. If the amino acid has different chemistry, you'll have an issue. And then lastly, we have nonsense mutations, which they change the amino acid to a stop codon. So this is really, really bad when it happens, because it essentially means that the protein after that point is just not going to get made because it's hit the stop codon, and so it's almost certainly not going to be a functional protein anymore unless the stop codon happened, unless this all happened to be at the very, very end of the gene sequence. But the odds of that are extremely low. So if it changes to a stop codon, if you have a nonsense mutation, it's very bad for the protein. Insertions and deletions are also really bad. They are when you either add or lose entire pairs of nucleotides in a gene. So you're actually either making the strand longer or shorter. This is, again, generally disastrous because it often, most of the time, causes frame shift mutations. So if I add or delete letters or if I add or delete bases that are not in a multiple of 3, what's going to happen is it's going to shift every single codon after the point of the mutation. So it's going to, no matter what, change every single amino acid that comes after it, which, as you can imagine, probably makes the protein non-functional. The only exception to this is if you add or delete 3 nucleotides, or 6 or 9, for example. You don't get a frame shift in that case because you're adding whole codons, essentially, but you will get either extra amino acids or missing amino acids, depending on if you're inserting or deleting, which will still likely have an effect and change the functioning of the protein. It's a slightly better chance that it'll still be functional, but it's still not a very good chance. Generally, insertions and deletions are very, very, very bad. Mutations do happen naturally, just from like mistakes in DNA repair and replication. Even just going outside, light from the sun will cause DNA breakages and mutations. So it is an unavoidable part of our life, but there are things that speed it up and increase the rate of mutations, especially as humans have become better at controlling like the world around them, essentially. Tanning beds are a really big source of UV radiation that can be very, very bad for you. Generally, tanning itself is just not great overall. X-rays, they have medical use, and it is tracked. The radiation doses you get are low. The bigger threat is actually to the people giving the X-rays to you. They will be exposed to a small amount, and that will cause additional mutations, so they are monitored. Smoking of any type, cigarettes, any type of smoking, doesn't matter what it is, can be sitting at campfires all the time, that will still cause increased mutations. Acne products, face creams, even things like HPV and other viruses have been shown to increase the rate of mutation. So if anything increases mutation rates, we just call them mutagens, which is a really cool name. So what are the effects? What's the overall point of all of this? Often, most of the time, it is no effect. For it to have an effect, it needs to be, first of all, in a gene, and then it needs to be at a certain spot in a gene, and then it also has to cause an actual amino acid change that actually has an effect. So there's a lot of things that have to happen for mutations to actually do something. But there is a benefit, and it is the raw material for evolution in the form of genetic diversity. Mutations do not drive evolution at all. They are much too random and slow to really drive mutation. Mutation is driven purely by variation of our already existing genes. So all mutations do is occasionally give us additional variation to draw on. Sometimes, though, it will benefit an organism immediately. It can give advantageous changes to your kids. It's just usually, for example, if my environment, if I'm a lizard and it's getting too hot outside, I can't just mutate to adapt to the cold. Generally, I have to have kids that will be better adapted, but it's not, individuals do not evolve, is the rule. The drawbacks of mutations is that you can pass on harmful mutations, and many often lead to cancer. You're more likely to have a bad effect from a mutation than a good one, as a general rule. So they're generally not helping us out. Occasionally, rarely they do, though. And that is actually the end of this, and I don't have a title card for the next one. Hopefully this one wasn't too long if I look at my time here now. Oh, under 10 minutes. good enough. I'm going to stop it at exactly 10 minutes just for